Hi everyone, my name is Kayla Curry, and today I will be talking about venereal syphilis. And I do apologize if you hear any loud noises. I live next to the highway and a club, so if you hear anything loud, just bear with me. Alright, so a little bit on the background of syphilis. It is a sexually transmitted infection that is spread by direct contact, such as kissing, sexual activity, and contact with sores or rashes of an infected individual. So some of the signs and symptoms include lymphadenopathy, which is swollen lymph nodes, splenomegaly, which is enlarged spleen, as well as headaches, rashes, and sores in the mouth, genital, or rectal areas. And a person may also develop fevers, chills, and muscle aches. So syphilis does occur in stages, and it will progress over time. In the primary stage, this is when a person will typically see rashes and sores and enlargement of lymph nodes, and this typically occurs on average 21 days after initial infection. So it has a pretty long incubation period. If left untreated, the symptoms could progress into the secondary stage, which is the development of skin manifestations such as pretty gruesome reddish-brown rashes and the onset of flu symptoms. Lesions will also form on mucous membranes of the mouth and genitalia. And a lot of growth happens in these areas because the permeability of mucous membranes are pretty high, and so a lot of nutrients could flow in and out. And secondary syphilis usually occurs several weeks after original symptoms have subsided. And this is a pretty big issue because if a person in the primary stage does not seek treatment and then their symptoms eventually go away, they may progress into the secondary stage and they'll develop these kinds of symptoms. Alright, so moving on to the tertiary stage, it is when the spread of the infection goes through the bloodstream into other parts of the body, including the eyes, heart, and brain tissue. And this could lead to possible paralysis as well as organ failure. Despite this being a pretty progressive disease, some people may not develop the tertiary stage, and this is because, for one, they either may seek treatment or their immune system was just able to fight off the infection. And tertiary stage usually occurs years to decades after initial exposure. However, a study done in a tertiary hospital in Japan, 8 men developed ocular syphilis. However, 7 out of the 8 of them developed these symptoms after 11 months. So some of the symptoms that we see in tertiary stage of syphilis, we may see those within just 11 months after initial exposure. However, these numbers are just relative to what we see on average. Alright, so some of the topics I will be talking about today is microbiology, so the study of microorganisms, immunology, which is the study of how the immune system reacts to these antigens, and biochemistry and biomedicine. So biochemistry being the combination of the fundamentals of biology and chemistry, and biomedicine, which is using the knowledge of biology to create medicine and treatment. Alright, so what causes the disease of syphilis is treponema pallidum, subspecies pallidum. And we're going to specifically talk about subspecies pallidum because treponema pallidum is able to cause many different kinds of diseases, not just syphilis. So for now, it's just going to be subspecies pallidum. And this causes they're made up of gram-negative corkscrew spirochete bacteria that causes periplasmic flagella, as well as inner and outer membranes that surround the appendages. They also have a unique motility of rotation and undulation of endoflagella. and they're able to survive and reproduce in narrow ranges of pH of 7.2 to 7.4, and in temperatures of 30 to 37 degrees Celsius, which makes the human body a perfect place to grow. They're able to reproduce via binary transverse fission, and they have a strong structural envelope and outer membrane that lacks LPSs, also known as lipopolysaccharides, but we'll get into those outer membrane proteins later on. They're also able to withstand high temperatures, and in a study conducted by scientists, they isolated these proteins by using Triton 100X. After isolation, they heated up these bacterial cells to 37 degrees for 30 minutes, and it showed no signs of denaturing of the proteins, which means that these cells are able to withstand pretty high temperatures and even fevers. All right, so immunology. The human immune system first deploys DHT after 24 to 72 hours after exposure, which is controlled by CD4 cells, also known as T cells. T cells produce cytokines that target pathogens and send signals to macrophages, which are phagocytes, as well as lymphocytes. So up in the right-hand corner, I have a video of a macrophage going to work. And so what they do is they'll use their appendages to reach out to these pathogens and engulf them. All right, so B cells, which are also part of the lymphocytes, are typically ineffective in fighting off T. pallidum due to the bacteria's unique outer membrane and lack of outer membrane proteins for their antibodies to bind to. So who's responsible for this is the TPRK gene, which encodes for the OMPs and gives the protein antigenic variation. The proteins on the outer membrane are also able to form beta barrels, which allow DNA and nutrients from the surrounding environment to be picked up and increase pathogenicity and genetic variation, as well as getting in an abundant amount of nutrients. All right, so bad blood. At the turn of the 19th century, there was a lack of treatment for bacterial infections, and there were little knowledge of how STDs and STIs were spread. Those carrying syphilis were burdened with the social stigma of having bad blood due to the development of tertiary syphilis. So, fun fact, Al Capone, as well as Vladimir Lenin, both died of syphilis in the early 19 to mid-1900s. All right, so some biomedical advancements. In 1906, the Wassermann test was created by August Paul von Wassermann, and this is in which he took a blood sample from a possible infected individual and isolated the liquid from the blood, which was called serum. And he created an antigen, which is made up of cardiolipin, lecithin, and cholesterol. He then mixed the serum, antigen, and sheep's blood together, and if there were no signs of hemolysis, then that person would be positive for syphilis. In 1910, the magic bullet was created by Paul Ehrlich, the father of chemotherapy, and this was salvarsan, which is an organoarsenic compound, and it's made up of a mixture of cyclopolyarsines, and this was the first use of chemotherapy. So in this compound in the bottom, you can see that there are 2 hydrocarbons as well as 2 arsenics, and this is neo-salvarsan. However, these cyclopolyarsines could have a many different number of arsenic. All right, so in 1928, Alexander Fleming accidentally discovered penicillin, which is a bacterial killing fungi, and this is used as a broad spectrum antibiotic, and it's made up of beta-lactams and a thiazolidine ring, and they produce enzymes that bind to the peptidoglycans in the bacterial cell walls and disrupts its overall structure. Due to these biochemical innovations, syphilis could be prevented from reaching secondary and tertiary stages. And in the United States, the mortality rate dropped from 10.7 to 5 in 100,000 from 1940 to 1950. And syphilis mortality rates do remain unknown in the early 1900s, but we can assume that after the discovery of antibiotics and the magic bullet, the number of deaths decreased significantly. And thanks to these biomedical advancements, syphilis now remains a relatively treatable and non-fatal disease. All right, so thank you all for listening, and if you have any questions, just let me know.